discuss them with you. So the first thing I always like to ask people when I am talking about microorganisms is, what comes to mind when you think about microorganisms? And I think that's a pretty varied background in terms of cultural, as well as sort of economic and social standing, and everyone sort of has a differing opinion, but there is some general commonalities to what people think about when they think about microbes. So if you want some extra credit points, drop in the chat what you think about microorganisms, and then by the end of class, if you want to drop another sort of line in the chat about how this sort of lecture has potentially changed your opinion about microbes or has reinforced your opinion of microbes, please do. Because I'm always very interested to hear what people's sort of attitudes are about microorganisms. And my guess is your attitude right now is relatively negative because your life has been completely disrupted by COVID. But, you know, hey, maybe you're like me and you love your microbes. So as we talked about last class, the earliest form of life on this planet was microscopic. And the Earth as a whole was dominated by microbes for a very, very long time. So remember, on this sort of biological clock, life arose sometimes around 9 minutes an hour. Microbes were all low until about 40 minutes. So 30 minutes, or about half the history of this planet, was just a microbe-only planet. And microbes, as you can imagine, and as we talked about with sort of the Great Oxygenation event and sort of other ways that microbes sort of set the stage for evolution, microbes were really, really, really important. To answer the question from Brianna, planktonic tunicates are microorganisms. The sort of broad definition of a microorganism, as we'll talk about, is anything you can't physically see with the naked eye. Microorganisms, in particular tunicates, those are what we call eukaryotic microorganisms, and that's sort of a distinction we're going to get into as well. So let me throw some fast facts at you. Let me see if I can blow your mind. Well, I don't know, maybe I can't blow your mind, but maybe I can try. So microbes outnumber all other species and make up most of the living matter on the planet. And what I mean by that is as I mentioned, there's about 15 million species of big things on this planet. We estimate there's anywhere between 25 million and 150 million species of microbes on this planet. And in terms of sheer biomass, microbes make up around 60% of the Earth's biomass. And in some ecosystems, they are the only biomass. For instance, if you look in the soil, microorganisms make up about 90% of the biomass in soil. And then you go to some more extreme environments, like say the bottom of the ocean, they make up 100% of the biomass. So microorganisms are super duper abundant, and they're super duper massive. Every time you go swimming in an ocean or the lake, you're being bathed in a sea of microbes. There are 1 million microbes in 1 milliliter of seawater, which means there are about 1 billion microbes in a liter of water. Or if you're in the gallon sort of system, there is about 4 billion microbes per gallon of seawater or lake water. So when you go swimming, you're literally swimming in a pool of microbes. If you could shrink humans down to the size of microbes, we would essentially be thinking about an espresso cup. So very, very, very tiny. We like to think there's a lot of us, but there's 7 billion humans on the planet. Right now, we estimate there's about 10 to the 30 microbes on this planet. So there's a lot of them. On your body, there varies depending on how big you are and how recently you have pooped, but there is anywhere between 30 trillion and 100 trillion microbes living on and inside you right now. And it's typically, again, depending on how recently you've pooped, there is about equal numbers or greater microorganisms inside and on you. So you are a microbial world. And sort of a weird way to think about this is they're equal parts you and microorganisms in your body in terms of cell number, which is sort of, I kind of think that's a weird way to think of it, but that's just me. And if we think about sort of biomass of microbes on your skin, they weigh, again, depending on the person, between 1 and 5 pounds. So 1 in 5 pounds of you is microorganism. So, if you weigh 150 pounds, well, you really weigh only 145 pounds because those microbes are making about 5 pounds of you. Again, there's a massive number of species on this planet. Most of them are microbes. And microbes really make life sort of happen. We're going to talk about this today, but they cycle nutrients, they create oxygen, they move energy around systems, and they do a lot for us. And so we're going to talk about all those wonderful things, but we're going to start off talking about the biology that these microorganisms do. So as I alluded to in previous classes, there are two sort of broad categories of cells that we find on this planet. We have the eukaryotes, and this is where we fall. This is where humans fall. This is where plants fall. This is where ants fall. This is where everything big falls. Anything you can see with the naked eye falls in this sort of eukaryotic category. Now, that being said, there are microscopic eukaryotic cells. So one of our classmates brought up plankton, and plankton, a lot of plankton are eukaryotic. And sort of the broad definition of a eukaryote, meaning that they have organelles. So you can see this sort of basic cellular structure here of a eukaryote. And you can see all these little sort of names being poked around here, right? So you're looking at the nucleus and the mitochondria and lysosomes and all sorts of different, what we would call organelles. This is this defining feature of eukaryotic cells, essentially specialized compartments inside these cells. And that's what really sort of defines a eukaryote. This is what defines you. You have all these specialized components inside your cell. And we're going to dive into these specialized components actually next week on Monday and Tuesday. And we're going to talk about what they work, how we put this all together, how your cells work as a whole. But we're going to start with the very sort of basics, talking about the microbes. So those are our eukaryotes. Most of our microorganisms are bacteria and the bacterial cousin archaea. They are prokaryotes. And so as you'll notice, the defining distinction between a eukaryote and a prokaryote is the lack of these organelles. You'll notice that a bacterial cell is very simple. It's just got its DNA. You'll see it's got lots of ribosomes. So remember ribosomes being the site of protein synthesis, as we talked about in the DNA lecture. And that's pretty much it. They don't have any organelles. And that's sort of how you broadly define a prokaryote. A lack of organelles or a lack of specialized structure. So anything that's going to happen inside of a bacterial cell or an archaeal cell happens sort of just in the open void of these cells, whereas opposed to anything that really happens in a eukaryote happens in one of these specialized components. So life is really divided into two sort of main groups. Our bacteria and our archaea fall into this category. Humans, plants, things like fungus, yeasts, they all fall into this eukaryotic. Eukaryotic cells, again, are much, much more complex. And this is, thinking about sort of early life, this would be more akin to what early life would have looked like. Just sort of a cell with some stuff inside doing some action. Sort of when we're thinking about microbe sizes, anything less than 0.1 millimeter is typically considered the cutoff for a microbe. So 0.1 millimeter is 100 microns or 100 micrometers or micrometers, depending on how you want to say it. And this is sort of the cutoff. The human eye really can't discern anything smaller than 0.1 millimeter. Typically, we can barely discern things, again, depending on your sort of eyesight, less than about 0.5 millimeters. So we really have a tough time distinguishing a lot of things. But sort of the cutoff when we're talking about a microorganism is 0.1 millimeters. That being said, there are exceptions to this rule. And so there's a really cool bacteria out there called Thiomargarita nambiensis, or the sulfur pearl of Nambia. It is actually the largest currently known bacteria that we know of. I'm sure there would probably be larger ones out there. And so this is what Thiomargarita nambiensis looks like. It's this chain-linked bacteria, and you can actually physically see it with the naked eye. And so this is a fruit fly. The scale bar here is 1 millimeter, and this is our bacteria. So this is an absolutely colossal bacteria. You guys have all seen fruit flies before. They are big. You can see them with your naked eye. You can actually see this bacteria with the naked eye. So it is pretty huge. But typically when we're thinking about a microbe, we're thinking about something you can't see. But again, there are, exceptions to this rule. When we're thinking of microbes, they're typically unicellular organisms. And microbes when we're thinking about them, it's typically bacteria and archaea. And we'll talk about the difference between bacteria and archaea very shortly. But again, as I mentioned, there are microscopic eukaryotes. And so typically, again, we're thinking about microbes being unicellular, being just one cell, as opposed to us, which is multicellular, right? And I've told you have trillions of cells that make up you. But there are some macroscopic unicellular organisms. So we talked about Thiomargarita nambiensis. And there are all sorts of algae and amoebas, which are microscopic eukaryotes, that are large. And so you can physically see a few examples of this. So we have two algae here. I'm not even going to try. These two algae right here. I'm a microbiologist, but I'm terrible at pronunciation of genus species. But what you can see is you have two algae here, and they look they're large, right? They look like they would be multicellular. But actually, these ones that you can physically see with your naked eye are actually just one giant cell, which is actually kind of crazy. If you want to sort of take a trip down crazy biology, this algae down here at the bottom is a really fascinating look at what happens when biology kind of goes crazy. So just sort of note, when we're thinking about microbes, it's typically unicellular, but there are giant things that are unicellular. And there are also many things that are multicellular that are microscopic as well. And so you can have multicellular fungi or algae or potentially microscopic animals like a dust mite or a rotifor or a copepod that are microscopic, but are multicellular. So it's sort of, when we're thinking about microbes, typically what we're thinking are things that are very small and typically things that are unicellular. But I just wanted to point out there are exceptions to this rule. Biology is a wacky and wild world. And I hope you guys have sort of already picked up on how bizarre biology is at this point, but you're definitely going to pick up on it going forward. So, as I mentioned, most of life's diversity is microbial. And so this is sort of just a very basic tree of life. And so what we're looking here is a phylogenetic tree. The idea being is when we're looking at this, the less branch length, so this sort of space here between two what we call nodes or tips, the less branch length you have, the more closely related these organisms are. So for instance, I have animals and fungi over here. They're very close, which means that animal and fungi are very closely related. Conversely, if I look at animals in, say, a bacteria over here, like spirochetes, so for instance, spirochetes, this is where the bacteria that causes syphilis falls in. You can see that there's a lot of branch length between these 2 points, meaning they're very distantly related. And when I say distantly related, I mean that in a phylogenetic sense, right? And I mean that as not just phylogenetically, I mean that as an evolutionary speaking. It's been a very long time since a spirochete and an animal have shared an ancestor, meaning something that they evolved and diverged from. And so when we look at sort of these three groups of life, there's our prokaryotes, which include our bacteria, which is most things you think about when you think about sort of microscopic things. And then we have our archaea, which is a sort of a hybrid sort of between bacteria and eukaryotes. As you'll notice on this tree, our archaea are actually much more closely related to us than they are to bacteria. And then we have our eukaryotes, which sort of includes all the big things, algae, plants, animals, things like that, but also lots of microscopic things like dinoflagellates and all these microscopic things and molds and fungi and sort of all these things that you sort of maybe have never heard of but are considered eukaryotes. But in terms of evolutionary history, everything on this planet at some point has shared an ancestor. And many times when we think about this, they're closer than others. So animals and fungi, for instance, have a much more, in terms of history of this planet, a much more closer ancestor as opposed to, say, animals and archaea, which our ancestor would have been a very, very long time ago. Now that's in contrast to what we call the last universal common ancestor, which is the sort of the last thing that we all descended for. So there was some organism way back in evolutionary time that sort of evolved in multiple different paths that led to the rise of bacteria, archaea, and eukaryotes. So this is what we call the last universal common ancestor, or LUCA. So this is a very simplified sort of tree looking at microbes. When we look at actual meaningful biological trees where we sort of plot millions of species on the same tree, you can see that the vast majority of life actually falls in the microbial world. So this is all our bacteria up here, this is our archaea, and this is our eukaryotes. So you notice that the bacteria and archaea side is much bigger than the eukaryotes. And this tree here doesn't quite get at that diversity because it kind of shows equal numbers here and here. But a real tree with all life on it really looks like this, with a microbial tree just exploding, being much, much larger than the eukaryotes, which are down here. But life is mostly microbial, and that intuitively makes sense, right? Microbes have been around here for a very long time, right? They were the very first organisms. They've been around this planet for billions of years. They were alone on this planet for billions of years. They've had a much longer time for evolution to take a foothold. And remember, new traits, new species arise through evolution, and evolution that takes period potentially over long periods of time. So the longer you've been around, the more time you have to evolve as a species or as a group of species. But that being said, even though they've been around for a very, very long time, and they are extremely successful groups of organisms, prokaryotes, which again includes bacteria and their archaea, have very, very simple cells. And this is going to be in direct contrast to what we talk about next class, where we talk about a much more complex cellular structure within our eukaryotes. Now, you'll notice that this very sort of basic microbial cell has what's called a nucleoid. This is just simply the region of its cell that the DNA resides in. So these have DNA, just like us. Remember, our bacteria only have one chromosome, or most eukaryotes, including us, have multiple chromosomes. They have what's called the cytoplasm, and we're going to talk about this more next week, but this is just basically an aqueous solution where basically everything in the bacterial cell happens. And they have their ribosomes, which is, again, where the site of protein synthesis. And then you'll notice they have a couple different sort of layers of their cell, right? They have a membrane, they have a wall, and some bacteria even have what's called a capsule. But that's it. That's really all these microscopic organisms have. They have an open space where they store their DNA, where they store their ribosomes, and they have some container around it, right? They have some wall, they have some cellular membranes, and that's pretty much it. That's pretty much all that consists of a prokaryotic cell. Now, there are exceptions to this rule. There are wacky and wild things that some bacteria have evolved because of their crazy metabolisms, but this is pretty much it. In addition, some bacteria will have what's called a flagella, or some bacteria will also have what's called a pili, and both of these are involved in movement. So a flagella is what sperm have, and so it's basically like a whip-like structure that you could think of like a tail, right? It propels them through a liquid solution. Pili do the same thing, but they're also involved in attachment. So if you ever get tuberculosis, for instance, what makes tuberculosis really, really hard to get rid of is actually these pili that attach to your lung cells. So they're involved in movement as well as attachment. Just as a sort of a fun fact. I don't expect any of you living in the United States to get tuberculosis, but there is a possibility. So in terms of differences between bacteria and archaea, they are two distinct groups of organisms, but they don't differ sort of meaningfully. They're pretty similar overall. They have different sort of metabolisms and different habitats and stuff like that, but the big thing that differentiates bacteria from archaea is actually what their cellular membrane looks like. And so just notice this is what differentiates. Bacterial membranes have slightly different structures in the membranes, and that's really the only meaningful thing that differentiates bacteria and archaea from one another. Otherwise, you can physically treat them as the same group of microorganisms. But just wanted to make that known. They are distinct, so phylogenetically speaking, again, they are distinct, archaea being more closely related to us than they are to bacteria. But there's not a lot structurally that differs between bacteria and archaea. And that is because they evolved from a similar common ancestor here. But the bacteria and the archaea keep it simple, right? Simple structures, huge amounts of biomass, but very important functions in this very simple structure. In terms of the genome of the bacteria, as we've talked about, it is just one single circular chromosome. This is the same thing for archaea as well. And this is, again, in contrast to eukaryotes, or including us, which have multiple copies of chromosomes, right? So if we're looking at this human set of chromosomes here, right, we have paired copies, duplicates of every chromosome we have. In addition, bacteria also have these what are called accessory chromosomes. They are called plasmids, and these are typically acquired through a process called conjugation, which we'll talk about shortly, which is basically a bacterial way of exchanging genetic material with one another. And these plasmids carry all sorts of important biological things, including like genes that help bacteria survive toxins, but they also carry genes that involve antibiotic resistance and virulence, which is the sort of concept of a bacteria becoming more infectious. And so while most of the DNA of a bacteria is in its physical chromosome, they can add these accessory plasmids that can make them much more sort of overall functional. Microbes, unlike us, as you will see next class and in the coming classes, reproduce by a process called binary fission. And this is in contrast to eukaryotes, which reproduce by a process called mitosis. And again, we'll talk about this in the coming lectures. So the idea being is a bacteria or an archaea, it's sort of just hanging around when it's sort of the conditions are right, when it's when things are, there's lots of nutrients around and things are sort of amenable to making the next generation of cells. What this microorganism will first do is it will make a copy of its single chromosome. So it as identical as it can make it. They make a copy of it, and then what they do is they start to split. So they essentially elongate their. Do anything particularly exciting here. They just go from one cell duplicating their genome to two cells that are identical, that have everything the bacteria needs. All those components in that cellular wall, in that sort of cellular component that I showed you a few slides ago, each daughter has that exact same component. And that's it. That's binary fission. It's really, really simple. It's been around for, we think, for a very long time. We think this is the sort of one of the original things that life evolved with this process of binary fission, and it hasn't really changed very much overall, which you know, I guess may be disappointing in one sense. Maybe you would expect them to get much more complex, but I think in a more realistic evolutionary standpoint, it sort of tells you that, well, it's really successful and it's really good. So why change what's really good? So microbes can do this process of binary fission extremely quickly, and so you can. There's all sorts of cool websites out there where you can do simulations of microbes, and this is where these ones are pulled from. We have a couple different. We have Streptococcus pneumoniae, which causes pneumonia, and you can look at sort of simulations of population size and the number of divisions that happen, and it happens really fast. And so the fastest dividing microbes that we know of can replicate and divide every 6 minutes. So you can go from one microbe to two microbes every 6 minutes. And so it's extremely fast. And this sort of rapid division, you can see, if you want to take a peek at this, I don't see the point of playing YouTube videos and stuff like that virtually, but I recommend taking a look at this video. It's sort of a real-time image of how microorganisms divide and how fast it is. It's absolutely astounding. In addition, it's sort of just one last thing I will mention is this sort of rapid division also occurs in pathogens. And so when we think about when you get sick, for instance, if you go to Chipotle and you get a burrito bowl and the lettuce is tainted with E. coli, you start experiencing symptoms within about 24 hours. Well, that 24-hour period is all it took for that pathogenic E. coli to divide and replicate and to produce enough E. coli inside your body to start making you sick. So this rapid sort of doubling, this rapid reproduction is what makes pathogenic organisms particularly hard to deal with, and how they can sort of spring up and like that. As we'll talk about later in the class, viruses do the same exact thing as microbes, as our sort of prokaryotes, in terms of rapid cellular, in terms of rapid reproduction. They just do it much faster, which is why you can get influenza or even something like COVID like almost overnight. So microorganisms, sort of a classical trademark of all microorganisms is very rapid cellular division. Microbes, while they do reproduce asexually, they can have sort of a form of sexual interaction with one another. And, you know, it's not like, you know, thinking about what humans do for sex. It's sex, again, as sort of a basic idea, it's just the exchange of genetic material, right? That's all sexual reproduction is made for. You're trying to exchange DNA, which helps the next generation. And so bacteria can have a sort of a modified version of sex where they essentially exchange DNA through this sex pilus. So one bacteria will essentially extend out this pilus, this sort of long tube, and they'll swap DNA. And the DNA they're most commonly swapping is these plasmids. And so this is how bacteria can directly exchange DNA. Again, it's sex, but it's not really sex, right? You know, sex involves sperm and eggs and all those different things. This is just a direct swapping of DNA. If you remember back from our class about evolution, this is what we call horizontal gene transfer. So it's not true sex, but people liken it to sex because it's an exchange of genetic material, which is again all real sex is for. And so microorganisms, they've been around for a really long time, and they do a crap ton for this planet. So when we think about what we do for metabolism, we sort of talk about it in very basic terms, right? We take in sugars and fats and proteins, we perform respiration inside of our cells, and we get energy from that, right? And remember that energy form that we're looking for is ATP, and that ATP form is the same, right? It's the same in microbes, it's the same in us, and it's the same as whales. But the way we get our food is, again, a heterotrophic way of lifestyle. But microorganisms, they've kind of gone crazy, right? They've had billions of years to evolve. They live everywhere on this planet. So they've evolved metabolic pathways that allow them to take advantage of multiple different energy sources, right? Again, us is just simply consuming, right? Plants are consuming light, right? Bacteria have evolved multiple different ways. And so bacteria can be autotrophic, meaning they can take in sunlight and expel CO2 like plants and trees do. They could be photoheterotrophic, where they take in sunlight to get energy, and then they use that energy from the sunlight to break down organic matter. So as opposed to using cellular machinery, they use sunlight to do it. They can be a chemoautotroph, where they take inorganic materials, so things that are not carbon, like sulfur or hydrogen, and they can use that to fix CO2 to make their own food. So they're still autotrophic, but they're using chemicals as opposed to sunlight. This chemoautotroph, if you remember back from last class, when I talked about anoxygenic photosynthesis, that's chemoautotrophy. And then finally, they can also be chemoheterotrophic, and there's a whole other way you can mix and match all these sort of different definitions. But the point being is that microbes, basically anything you can think of on this planet for an element, a microbe has evolved the way to use it in their metabolism. And I actually mean that. There's any metal that you can think of, aluminum, something like is crazy like uranium. They've evolved some way to use that in their metabolism. And so just the whole point of this slide was to show you that microbes, while we are very simple metabolically and very, very complex sort of cellular level, microbes are the exact opposite. They are very complex metabolically, meaning they can do a lot in nature, but they are very simple at the cellular level. So microbes have sort of gone the opposite route of us, right? They've evolved crazy new metabolisms, crazy new ways to live in environments, as opposed to making their cellular structures complex. Which I think is a important thing to make note of, is that even the microorganisms inside your body and on your skin, they've evolved all sorts of crazy ways to metabolize things. And we actually take advantage of all these crazy metabolisms inside of our body to get more energy from our food. But that's a topic for another day. Microbes as a whole in nature are sort of the half of the picture when thinking about how we cycle carbon. So anytime you hear this idea of a cycle in nature, it's basically how things are moved through different organisms. Because when a tree photosynthesizes something, well, it produces leaves, and eventually those leaves die. And those leaves just don't sit there. They get decomposed. They get cycled back into the system. So anytime something is sort of used by biology, there's some sort of associated cycle with it. So if it's carbon, well, we have the carbon cycle. If it's nitrogen, it's the nitrogen cycle. If it's phosphorus, it's the phosphorus cycle. If it's uranium, it's the uranium cycle. So there's a cycle for everything, because nothing in nature gets wasted, because there's always something sort of waiting around the corner that could potentially use it. So the idea being is microbes play a really key role in this cycling of carbon. And remember, carbon is the second most abundant element in your body. Everything on this planet is carbon-based, so everything participates in the carbon cycle. The idea being we have primary producers, which are our plants, which are our bacteria, right? They're taking CO2 from the air, they're making sugars. And this carbon that has been fixed, that it's been photosynthesized by these organisms, gets consumed by bigger things, right? By insects, by, rabbits, so on and so forth, which are then consumed by other things, which are then consumed by other things. And eventually, everything either dies. And so once things die, they are then sent back into the ground, where they're eaten by bacteria and fungi, which recycle all that carbon back into the system. Or these organisms, while they're alive, are producing CO2, which is then consumed by the primary producers. So this is why it's called a cycle, because it sort of just does this cyclical thing. Everything is breathing CO2, everything is eating something, everything is dying, everything is being sort of worked its way back into the system. And as you'll notice, bacteria form sort of two key links here. They're responsible for this sort of recycling of dead stuff back into sort of the biological system. But they're also responsible, again, for a huge proportion of the primary production or the photosynthesis that occurs on this planet. And they do sort of all this sort of carbon cycling and all these metabolism in extreme environments. And so I'm going to sort of, maybe I won't blow your mind, but I'll show you some crazy stuff that microorganisms do and where they live. And so there's bacteria that we know that have been isolated by researchers from the University of Pennsylvania. They found this bacteria. I'm not even going to try to say its name, but it was recovered from ice that was 2 miles beneath the Greenland glacier. All signs pointed it to be very, very old. So it was about 120,000 years old, this one microorganism they isolated. It was in a state called dormancy, so it was basically asleep. And so they found this microbe just kind of hanging around in the ice. And when they brought it back up to the surface, they gave it some water, they defrosted it, it came back to life after 120,000 years. In addition, they find microbes in the Atacama deserts. For those of you that have never heard of the Atacama Desert, it's the driest place on the planet. So for instance, this is where NASA sends its astronauts to go prepare for missions to Mars, because it's very Martian-like, it's very dry. It gets about 2 or 3 millimeters of rain every year. But that being said, there are microorganisms that are there. And so some scientists in 2003 found a very robust group of community of microbes, including this bacteria called Deinococcus. I'm not going to try and say its species name, but Deinococcus. That Deinococcus name should ring a bell. We talked about Deinococcus radiodurans in the lecture about DNA. And this is Deinococcus is a really hardy genus of bacteria. But it lives in an environment where basically there's almost no water. And so it survives in this extreme condition. Next up, we have a bacteria called Halobacterium NRC1, and it's found to be the most radiation-resistant bacteria on Earth. It could withstand up to 18,000 grays of radiation. A gray is one way to measure the amount of radiation that something receives. Just to put that in perspective, 10 grays of radiation can kill a human, so this bacteria can survive 18,000 grays, which is 1,800 times the amount of radiation we can, which I think is pretty crazy. So basically, you could find this bacteria hanging around after a nuclear bomb. So even if we wipe each other out with nuclear bombs, there'll still be microorganisms kicking around on this planet. We have another bacteria, whose name I can pronounce because I've worked with this bacteria before. It's Ferroplasm acidophilum, and this is a bacteria that they found in these acid mine drainage sites. So when they mine for gold and silver and all that fun stuff that we need for metals, they flood them with acid to help break down the rocks, and you get these acid mine drainages that leak out. And as you can see, they are gross looking. They completely destroy the environment. So any metal mining is inherently very damaging to nature, but inside these streams you find ferroplasm, and this bacteria can survive in pHs less than 1. And if you remember back from our pH sort of discussions, pH of 1 is very, very acidic. The pH scales is a 0 to 14 scale, and so this bacteria basically survives at the very end of the pH scale when life basically shouldn't be able to survive, but this bacteria still does. So it's a pretty amazing microorganism, and it's actually very pretty under the microscope, so just as you know. In addition, we find bacteria living in salt flats, and so for those of you that have never seen salt flat in person, but Maybe you've seen on pictures of it, or you've heard of the salt flats. But these are these hypersaline environments. They are basically just salt on top of salt on top of salt. And of course, there are microorganisms living there. And so, for instance, we have this Haloquadratium wasabi. It's an archaea, and it takes this sort of rectangular shape, and it uses this rectangular shape to actually survive in these hypersaline environments. So whereas if we took even a scoopful of this and we tried to eat it, we would likely succumb to it. This bacteria thrives there. There are billions of bacteria like this one in these salt flats. So, and sort of moving to away from crazy environments into sort of deep environments. We know there are bacteria that live at the bottom of mine shafts, and we know there are bacteria that live miles down in the Earth's crust. But one particular bacteria, Dosulfuritis, I'm not going to try. This bacteria here was discovered in a South African mine shaft, and it was about 2 miles below the Earth's surface. And what's sort of interesting about this bacteria, it was all by itself. There's no other bacteria that we could find around it. And what it was doing, it was actually using the radioactivity from uranium and using uranium in its metabolism to derive energy. And so this bacteria was its only sort of, it's one of the only known single species ecosystems. And if you want to read about this, I linked a link to Wire here. It's a really sort of crazy bacteria to sort of read about. In addition, we have microbial taxa living in the extremes of the ocean. So Pyrodictum abyssi is a bacteria we find in hydrothermal vents, so very deep, miles underneath the surface. It lives in pressure that would essentially pancake you. It would pancake even a submarine. And they are able to withstand this massive pressure. In addition, they live near hydrothermal vents. And so for those of you that have heard about a hydrothermal vent, you should know that they're hot. They are very hot. And you're talking about, essentially magma and volcanic activity leaking out of these vents. And these bacteria live right around these vents, sometimes even living in the plumes of smoke. And so they are able to withstand the extreme pressure and also able to withstand the extreme heat. And so this is what just as what it looks like. You have these hydrothermal vents. They have seawater that has all these minerals, and these bacteria actually take advantage in a process called chemosynthesis. So similar to those anoxygenic photosynthesizers I mentioned, where they take hydrogen sulfide, which is rotting eggs, water, and CO2, and they use that to make carbon, to fix carbon. And so they sort of make a living on all the sort of the things that are coming out of these black smoker vents. They also live in these crazy symbioses around black smoker vents, and in particular, they live with these worms. So they've formed a symbiosis, which is simply an association with another organism that's not the same species, and they live inside these worms that can be over 2 meters long. And these worms, they're white, and they got sort of these orange polyps at the end of them. And these worms are particularly happy in the environment. And this is what these worms look like. They're just tubes. They have these gills where they take in and exchange gases and water and all that fun stuff. But inside these organisms, because there's no sunlight down here, they harbor bacteria, and they harbor our bacteria that we were just talking about, and they harbor millions and billions of these bacteria that basically fix carbon for these worms. And essentially the worms are exchanging a safe place to live and sort of the sucking in of fresh nutrients with food from these bacteria. So they're forming this symbiosis. And so these little organisms power these monstrous 6-foot worms. And if that's not weird to you, then I don't know how much other weird stuff I can tell you about biology, but it's a pretty weird thing. 6-foot worms powered entirely by bacteria. There's needs nitrogen, and microbes are responsible for nitrogen. And so, for instance, microorganisms living in soil, they live in the ocean, they live in association with plants. They do a process called nitrogen fixation. Oops. And the idea being is they capture nitrogen from the atmosphere and they convert it into essentially fertilizer. And prior to the advent of the Haber-Bosch process, which occurred in World War I and World War II, the only way we could bring new nitrogen into a system was through bacteria. So we needed bacteria to supply all the nitrogen. So when we were growing crops before 1913, and well, World War II is when it really took off, but before 1913, we relied solely on the bacteria in soil to provide nitrogen for our crops. Sometimes we would add cow poop and stuff like that, but we really relied on the bacteria to do that. Fast forward to, again, 1913 and World War II, we invented the capacity to make our own fertilizer. So we thus removed the need of bacteria to supply nitrogen for our crops. And we solely relied on fertilizer. And you can actually just look at this, how many teragrams of nitrogen there are. A teragram, if I'm not mistaken, is 10 to the 15 grams of something, which is massive. And you can look at how the amount of nitrogen in the biosphere or on the planet has increased over time. So you can see that the total amount of anthropogenic or human-made nitrogen has just expanded. And you'll see that nitrogen produced via sort of our crops and things like that has sort of just steadily increased. So the amount of total nitrogen we've sort of put into the atmosphere, we put into the biosphere is massive. And there's sort of a natural range here, but as you'll see, by the time the 70s rolled around and we were dumping tons and tons of fertilizer into nature, it's just sort of a cliff that's naturally found for nitrogen on this planet. So, fun stuff. And again, microbes, they are responsible for that nitrogen cycle, and the nitrogen cycle is a really complex cycle. This is one of the things I was very interested in when I was a student. And there's all sorts of different processes involved here. And, you know, there's different processes that happen in nature, including denitrification and nitrification that we're not going to talk about. But these are all mediated solely by microbes, and this is an important way to process nitrogen in the system. So again, it's all mediated by microbes, including this really cool bacteria called Trichodesmium, which this is actually a picture I took when I was down in Bermuda. And you can see this microorganism forming clumps. And this microorganism, Trichodesmium, is actually fixing nitrogen and being actively involved in this nitrogen cycle. So again, nitrogen being the highly important thing to every life on planet. And without microbes, the cycling of nitrogen, the sort of movement of nitrogen through the ecosystems wouldn't happen. And without these microbes, we couldn't have trees, we couldn't have grass, we couldn't have anything. So without the microbes moving nitrogen, without them producing nitrogen, life as we know it wouldn't exist. In addition, microorganisms, as I mentioned, are responsible for you. And so we have a class later in the semester about this. We're going to talk about the human microbiome in our very last week. Because it's a very sort of complex topic with a lot of really interesting things that deserves its own thing. This is also what my PhD is in, so that's why we're doing an entire class on it, because this is what I'm an expert in. But we'll talk about this later in the semester. But just to know that the way you are as a person, you couldn't exist without microbes. You couldn't digest your food properly. You would get sick all the time. You couldn't produce half the vitamins you need to survive, including vitamin B, vitamin K. So you would be sort of, without microorganisms living on inside you, you'd be in a pretty sad state. So just as sort of a foreshadow about how microbes are important for you, they are, again, essential for our human life in terms of making you who you are. But as some of you, well, actually, none of you said. I'm surprised. So I was looking at the chat just now, and none of you said that microbes make you sick, which is I'm a little disappointed. That's usually the first thing people say. But microbes can make you sick, and that's sort of how we best know microorganisms. So much of microbiology and all the things I've sort of told you, we really know because we were trying to study microbes that made us sick. And historically speaking, death by microbe, whether it's a virus or bacteria, was sort of the most common way that people died. You know, you can think about massive events like the bubonic plague or the Spanish flu earlier in the 20th century. That was sort of one of the big causes of death, was microbe. And as we've evolved, as we sort of changed as a species, as we've gotten smarter and more scientifically advanced, including the development of vaccines, including the advent of sort of better medical care, better surgery techniques, and antibiotics in particular, we've sort of driven down the amount of deaths by microorganisms, but they still are a major cause of death in the United States. So for instance, about 18.3 million people die every year from infectious diseases. And if you sort of tally up the population of the 5 largest United States cities as of 2009, I got to update this. I like this slide because it's really cool, but that required me to go research all these numbers again, which I guess isn't that long. But anyways, I digress. As of 2009, the population of the 5 largest U.S. cities was about 18 million. So basically, every year on this planet, the population of New York, Los Angeles, Chicago, Houston, and Phoenix basically kicks the bucket from a microorganism. And so they are pretty abundant in terms of how much they kill, but they actually are dwarfed relative to other causes of death, like cancer and heart disease and things like that. But just to know, there are still millions of people every year who die from infectious diseases. And it's not an inconsequential thing, right? And you can also think of something like COVID-19, right? It's infected millions of people, and in the United States alone, 140,000 people have died from COVID-19, and that's just in the 5 months since this sort of pandemic has taken off. And as of now, we're looking at well over 2 million people dying here in the United States alone, unless we get a sort of effective vaccine. So that can be a pretty tough thing to take care of, and they can be a pretty deadly force throughout time. But since the advent of antibiotics, we've had a pretty dramatic decrease in the amount of deaths from microorganisms. And so this is looking at deaths per 100,000 people per year, and this is time on the x-axis. And what you'll see is that, it's been slowly decreasing over time. And part of this decrease over the time has been an increase in sanitation. So being clean, washing our hands, doctors being cleaner in their techniques and stuff like that, but also just not like dumping your feces on the street. It's kind of a big deal for humans. I know that's sort of a thing we don't really think of, but that was sort of the way that they did things historically, just sort of leaving your poop out and people getting sick from your poop that you leave out, which is just disgusting, but I digress. And you'll see that, again, the sort of the overall trend is downward, but you can see spikes, including the 1918 influenza panic, better known as the Spanish flu, which killed, about 50 million people globally. So you see that huge spike. But you'll see that once we started adding in antibiotics, so including sulfanilamide as well as penicillin in the 40s, you'll see sort of this dramatic drop that followed them. And so we really start to see, again, a pretty dramatic drop of in terms of deaths from pathogenic organisms by the advent of antibiotics, because antibiotics are great. They help your body rid you of an infection, and so they're a really powerful way of getting rid of microorganisms that are causing you to be sick. And then we've actually started to see an uptick in the mid 80s with the sort of proliferation of HIV and AIDS, and we're going to start. This is actually predicted to go up, and this is going to be predicted to go up because of antibiotic-resistant microbes. And so many of you, I hope, have heard of this concept before, but antibiotic resistance is a big thing in the medical field. And what this refers to is simply as we use an antibiotic more and more and more, as great as they are, microorganisms will evolve the capacity to be resistant against them. And they do this evolution in one of two ways. They either acquire resistance from a bacteria that already has resistance. So nature is chock full of antibiotic resistance genes because these antibiotics, chloramphenicol, tetracycline, vancomycin, and so on and so forth, they were naturally in nature. So all these antibiotics are microbial byproducts. And so in nature, these antibiotics already exist. And because they already exist in nature, there is a natural reservoir of genes, DNA out there that makes an organism resistant to these antibiotics. And so microorganisms can pick up these resistance genes from nature, or they can evolve their own resistance genes. So if they have a gene that's, say, resistant to penicillin, and they encounter ampicillin, well, these are the same class of antibiotics, and thus the way to sort of detoxify yourself from these is very similar. So slight changes to the genes that make you resistant mutations to the genes that make you resistant to penicillin could potentially make you resistant to ampicillin. And so this is an active thing. This is something we've sort of seen throughout the history of antibiotics, all the way back from the 30s up until now. And what you'll notice is that when we introduce the antibiotic, the clinical antibiotic resistance is observed very shortly thereafter. And so, for instance, we introduced penicillin in the 40s. By the late 40s, we had penicillin-resistant bacteria in clinical settings. And what you'll notice is that pretty much every antibiotic we introduce, well, antibiotic resistance follows pretty shortly after. And it takes sometimes a long time. So, for instance, something like erythromycin took about 30 years to evolve resistance, but that's because erythromycin is used very sparingly. Other antibiotics, such as vancomycin, took, again, about 30 years, but that's because vancomycin is an incredibly strong antibiotic, and it's something that takes a long time to sort of get resistant to. And so, but other antibiotics, such as, again, penicillin, only took a couple of years. And so the more we use antibiotics, the more the bacteria become resistant, and the more they use clinically, the faster we observe resistance. So, for instance, daptomycin was heavily used clinically because it was sort of a new antibiotic that we discovered, and within 2 years, we observed resistance in a clinical setting. So, as you can imagine, this is a big deal, right? We sort of saw the downward trend because of antibiotics, right? Being clean when you do surgery or being clean in your everyday life can only make you so less susceptible to microbes. Antibiotics really help us sort of stop infections. And you can imagine this is going to be a huge problem. And in fact, it is. It's sort of one of the sort of greatest threats to world health that we're observing, antibiotic resistance. And we'll get back to this in a second, but I want to just sort of talk about how microbe evolution fits into this sort of antibiotic resistance. And microbes typically evolve quickly. And this is simply just due to how they live in their everyday lives. They have very quick generation times. Remember, I told you some microbes can produce offspring every 20 minutes. They have very small genomes, and they have much less developed cellular machinery. In particular, the machinery that helps them repair DNA. So microbes mutate faster, which means they can evolve faster than us. And to sort of put this in perspective, you can look at the number of unique enzymes on the x-axis, the y-axis here, and time on the y. And what we're looking at is here what are called beta-lactamases, which are enzymes that detoxify groups of antibiotics called lactamases. And what you're looking at is here's the number of enzymes essentially identified during the age of antibiotics. The idea being is you're seeing that the number of unique ones that we're detecting, again, genes that make them resistant to this group of antibiotics, you're seeing that they just sort of take off, right? We have exponential growth, really fast growth. So these things are taking off, which really tells us that the evolution of these things is very rapid. So as we increase the number of unique ones, it means that they're evolving, which means they're mutating, they're changing. And you're seeing it's happening in a very short period of time, 30 years. So we're going from basically 0 up to 800 in 30 years. That's rapid evolution, very rapid evolution. And if you want to see how fast a microorganism can evolve in terms of antibiotic resistance, please watch this video. It is absolutely astounding to watch this. So this is a video. It's from a gentleman at Harvard named Michael Boehm. He studies antibiotic resistance in the clinical setting. And he did this really amazing experiment where you can see microbial evolution to antibiotics in real time. And so please, and one more time, please, take a look at this video because this is a really important topic. You might not ever think about antibiotics and antibiotic resistance, but it is a really important topic. So please take a few minutes after class and watch this video. It is crazy, but it is also super duper important. So please watch this video. And I want you to watch this video because we are entering what's called an antibiotic apocalypse. We're entering a period of time. Antibiotics are essentially going to be ineffective against most common human pathogens. So things like E. coli, things like Pseudomonas, things like Enterobacter, all sorts of very important clinical bacterial pathogens, our antibiotics are essentially going to be useless against them. And so we are entering this, what we call this post-antibiotic era, or this antibiotic apocalypse, that has sort of been a result of clinical bacteria evolving or acquiring antibiotic resistance. And the idea being this this antibiotic resistance and sort of just this trajectory that we're seeing here in this graph, again, all derive from overuse of antibiotics, using antibiotics a lot in a clinical setting. And because we've used antibiotics a lot and we've overused them in many, many cases, we're entering this antibiotic apocalypse. And just to sort of show you sort of an example of this, so we can look at hospital stays in a Pennsylvania hospital, or a group of hospital. And so this is MRSA, so methicillin-resistant Staphylococcus aureus, or MRSA. Many of you have probably heard of MRSA in the news because it's a really nasty bacteria. It infects your skin. And this bacteria is resistant to methicillin, which is one of the strongest antibiotics we know. And it was the primary way we treated staph infections, or infections from Staphylococcus. And so what you'll notice is that we have the number of discharges here from the hospital on the y-axis, all the way from 0 to 400,000, and the time on the y-axis. And you'll notice that this sort of trajectory of patients from this, again, this one hospital in Philadelphia, or this group of hospitals in Philadelphia, is essentially starting to take off exponentially. And what you'll notice is that it's happening pretty quick, right? 1993 to 2005. And this trend does continue upward. So we're seeing more and more antibiotic-resistant bacteria occurring over time. And this is a couple reasons for this. The first, we're using antibiotics too much. And so a lot of people will go to the doctor, and as a sort of cautionary measure, they'll get antibiotics when they really don't need antibiotics. The second thing we're seeing is actually people getting treated for bacterial infections but they have a viral infection. And so antibiotics only work on bacterial infections. They do not work on viruses at all. So if you have the flu, no amount of antibiotics you take could help you with the flu. If you have COVID, no amount of antibiotics you could take will help you with COVID. So antibiotics only treat bacterial infections, and we've been using them to try to treat viral infections. In addition, there's always this sort of pressure on the medical community that if you go to the doctor, you should get some sort of prescription to take care of your thing. And one of the things that doctors have been using to do this is by giving people antibiotics. And again, by overprescribing them, it's making more and more bacteria resistant to them. And then sort of finally, the way we're seeing it sort of, well, one of the ways we're seeing an explosion of antibiotic resistance is actually by giving it to livestock. And so there's this weird phenomena in agriculture when you feed cows and other livestock antibiotics, they grow faster, they grow bigger, and that overuse is actually proliferating antibiotic resistance in the clinical setting as well. And sort of to pair with this, there's very few drugs in development right now. It is not economically viable for a pharmaceutical company to make an antibiotic. It takes a long time to do, and they don't make very much money off selling antibiotics. However, they do make a lot of money off of selling, say, cancer treatments, or even selling things that, say, that treat erectile dysfunction. There's just not a lot of money to be made in making antibiotics. Thus, there's very from one of these bacteria, there's a high probability you might die. And it also will make simple medical procedures almost impossible. So think about when you go get surgery, you are almost always prescribed an antibiotic after the fact to prevent any potential infections that you might get. Well, think about if you have open heart surgery and you can't take antibiotics because they're no longer effective against clinical bacteria. Well, you might not die from your heart surgery. You might die just because you got an infection that couldn't be treated. And so this is a really sort of impending, looming apocalypse. I don't want to be sort of all doom and gloom about this, but I do feel like I should inform you about this in terms of this, because this is a pretty big health crisis that we're sort of stumbling upon. And it was totally preventable, but we're not good at preventing things, unfortunately. So, but again, there is an antibiotic crisis that is looming, and it's, again, from use and overuse of these antibiotics. But that's antibiotics. Let's sort of switch gears and talk about something that's on your mind every day, and that's viruses. And these viruses are pesky, and they are tiny. If you thought microorganisms, if you thought bacteria were tiny, viruses are even tinier. They are ultra small. And viruses are essentially parasites. They essentially hijack the host to make more copies of the virus, and that's it. That's what viruses do. They hijack you, they make you make more copies of them, which then lets them infect you more. But viruses, again, are parasites. And so viruses they take a wide variety of shapes and sizes and genetic compositions. And so you can sort of look at some cartoon illustrations of all these different types of viruses. So you all have probably heard of Ebola from the previous administration. The sort of the outbreaks that occur in Africa, hepatitis, rabies, influenza, all these different types of viruses out there, including a classification of viruses called bacteriophages, which look like crazy, I don't know, like they look like lobsters with or not lobsters, like crabs combined with like weird alien heads. I don't have a good way to describe that, but bacteriophages, which are viruses that only infect bacteria. And these viruses take a huge variety of shapes, and they have a huge variety of hosts that they can infect, and they are super duper abundant. I mentioned earlier in this class there are 1 billion bacteria in every liter of seawater or lake water. There is 1 trillion viruses in every liter of seawater and every liter of lake water. So they are even more abundant than bacteria. This is the most abundant group of organisms on the planet. And so viruses, like bacteria, are very, very simple. They have this sort of very basic structure. And so this is actually COVID-19, a cartoonized version of what COVID-19 looks like. And so what you'll notice is that these viruses have an envelope. They have this membrane that separates them from nature. Inside this, they have their genome. And this genome is one of the few ways that viruses are variable. Because viruses can have, unlike us and unlike bacteria, which just have a double-stranded DNA genome, viruses can have all sorts of crazy types of genomes. They can have a single-stranded DNA genome, a double-stranded DNA genome, a single-stranded RNA genome, or a double-stranded RNA genome, and there's all sorts of other combinations that I'm not going to get into. But viruses can have a very sort of diverse style of genome. In the case of COVID-19, very much like all viruses like it, has an RNA genome. It's a single-stranded RNA genome. But what you'll notice is that it's very simple. It's got a genome, it's got some sort of membrane, and then it's got these little proteins that are called spike proteins. And viral spike proteins, they're basically involved in attaching to their host. Because remember, all a virus does is infect a host. That's it. And I will also note that viral genomes are very, very tiny. Most viral genomes have less than 200 genes. And so, for instance, one of the most deadly viruses that we know of, HIV, has only 32 genes on its genome as a whole. So this really destructive organism has only 32 genes. And so you don't need a very big genome to be really destructive. Viruses have two very distinct life cycles. They fall into what are called the lytic and lysogenic cycles. And the way this works is a virus, if it's, say, introduced to its host, in this case we have a bacteriophage infecting a bacteria. The way viruses work is they attach to their host. They're essentially adsorbed to the host, not absorbed. They inject their genomic material, so their RNA or their DNA. What this genetic material does is it incorporates itself into the chromosome of the host, so whether it's my DNA or a bacteria's DNA. What they then do is reprogram the host to make more copies of the virus, all the little bits and pieces of the virus. They force the host to assemble those pieces into full viruses, and once they've assembled, varying amounts, so some viruses make a few hundred new viruses. Some viruses make 10,000 plus new viruses. But again, all forcing the host to do this. They essentially kill the host cell, rupturing and releasing the viruses, again, 10 to upwards of 10,000 new viruses, to then go on and infect more of the host. So if you think about how this happens inside your body, you get an infection, say in your lungs, it attaches to your lungs, it injects its DNA into your lungs, reprograms your lung cell to make more copies of the virus, then it kills your lung cell, sending out thousands of new viruses to infect all the lung tissue around it. And that's how a virus works. And this happens pretty quickly. You can have a virus, you can have a cell produce new viral viruses within 6 hours. So you can make tens of thousands of viruses in a very short period of time. So it happens very, very quickly. And so if you have the cold, if you have COVID, if you have something like HIV, this is what it's doing to you constantly. It's infecting you, reprogramming your cells, killing your cells, releasing thousands of viruses to then go on and infect more of your cells. Which is why you can go to sleep feeling great and wake up the next morning with the flu or the cold, because this happens really, really fast. So that's the lytic cycle. That's what these viruses are doing when they're infecting hosts. There's a second cycle that these viruses do, and it's called the lysogenic cycle. And this has some implications for some real-world diseases as well, but it's a relatively common life cycle. So the idea being is you have a virus infects the host, it injects its genome into the host, incorporating its genome into the host genome, and instead of reprogramming the host right away to kill the host, to make more viruses, the viral DNA just kind of hangs around. And it can do this for essentially indefinitely. So if you remember back from our DNA class, I mentioned that the human genome is about 7 to 10% virus. This is how your genome is 7 to 10% virus, because we got infected with viruses in the past and their DNA just kind of hung around in our genome. It never did anything. The virus just kind of hung around. Its DNA is there, but it's not infecting you. And so the lysogenic cycle is how you can acquire new DNA from viruses, but not be killed by the virus. However, the lysogenic cycle can persist indefinitely, but it can switch to a lytic cycle. And so the idea being is if you encounter some sort of stress, say like you get run down or you get sick with another virus or sick with another bacteria, the virus can switch from being a lysogenic virus to then being a lytic virus. So the best example I can give you of this is the herpes virus. This is the best way this has been studied. And so if you get infected with someone by herpes, the virus will inject its DNA into you, enter the lysogenic cycle, and then the virus will just kind of bide its time. It'll essentially be a jerk. So for those of you that ever have had like a cold sore, you know that this virus pops up when you get stressed. When you have say you have a big interview, say you get, you pull an all-nighter studying for an exam. Any sort of stress to your body can activate this virus. So it'll switch from being in this lysogenic cycle to then switching gears to being the lytic cycle. And so viruses that enter this lysogenic cycle can again stay indefinitely inside you and never cause any harm, but some of them can essentially wake up and start to cause an infection. And if they don't kill you after they enter this lytic cycle, they will then re-enter the lysogenic cycle once your body sort of gets a hold of the infection, just as a note. So if you get something like herpes or even the cold sores or something like that, it's something that you live with for the rest of your life, because the virus sort of flip-flops between being lysogenic and being lytic. But more importantly, you could also be infected with herpes and never know it, because your virus always stays in the lysogenic cycle. So just a fun fact of how herpes works. But as I mentioned, there are two distinct types of viruses. We have just sort of viruses, and these are the ones that infect eukaryotes. And then we have bacteriophages, which are the ones that only infect bacteria. And these viruses are teeny tiny. And so if we look at sort of a log scale of size, we have humans at, say, 1 meter plus, eggs at 100 millimeters, an egg of a frog at 1 millimeter, and then we start to get into things that are microscopic, right, at that 100 micrometer or 0.1 millimeter mark, and that's plant and animal cells. We get smaller to about 1 micrometer. That's our bacteria. That's where our bacteria sort of fall, that 1 micrometer. As we get into the nanometer sizes, well, that's where we start to see our viruses. So at 100 nanometers, that's about the average size of your average everyday virus. To put that in perspective, the flu virus is that big, COVID-19 is that big, HIV is about that big. So most viruses that we know are about on that 100 nanometer size. And then if we sort of keep going down on the scale, 10 nanometers is about the average size of a protein. Lipids that you can ingest are about 1 to 10 nanometers. Atoms are less than 0.1 nanometers. So very, 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 very tiny. What you notice is that viruses are small. That being said, not all viruses are small. As I told you, I like to show weird things, and there are giant viruses out there. And so these giant viruses, if we put them on a scale, they are about, well, it depends. They are in between bacteria and our cells. So they can be upwards of 100 microns wide. And they can be, they're generally around like 50 microns wide. So they're huge relative to your average virus. Again, essentially 100 times bigger than your average virus. And these giant viruses, they sort of straddle the line between, are they alive, or are they viruses, or are they eukaryotes? And you find them in water, and they affect protozoa, which are microscopic eukaryotes. And they have very, very large genomes. They have well over 1,000 genes in their genomes. And just sort of flash back to a few slides ago, remember, most viruses have a few dozen genes in their genomes. These viruses have well over 1,000. So they have more genes than some bacteria do. So they're huge. And they are bigger in size than the vast majority of your bacteria as well. But viruses, they play an extremely important role in nature. So we sort of talked about infectious viruses, but viruses are everywhere. I told you there are 1 trillion of them per every liter of seawater. And because they are so abundant, they play vital roles in the cycling of nutrients across the planet. So just to give you some numbers about how many viruses there are right now about 10 to the 28 bacteria in the global ocean. And to put that number in perspective, that means there's about 100 million times more bacteria than stars in the universe. So yeah, that's a lot of bacteria. There are 10 to the 31 viruses on the planet, and there's 10 to the 30 viruses in the ocean. I'm sorry, 10 to the 29 viruses in the ocean. So if you do the math, that there, in terms of viruses infecting bacteria, there every second in the ocean, the global ocean, if you looked at it as a whole, there are 10 to the 23 viral infections in the global ocean every second. Think about that, 10 to the 23 infections every second in the global ocean. And because there are so many infections, we have a lot of bacteria dying, which means there's a lot of new material being released into the ocean, which means other bacteria can eat that material and cycle all those nutrients, that carbon, that nitrogen, that phosphorus. So viruses do play a very important role in nature as well as sort of a clinical setting. And in terms of a clinical setting, viruses have been deadly. So some of the most deadly pandemics that we know of have come from viruses. And so we know that the Spanish flu being the most deadly of all, and so in 1918, between 50 and 100 million deaths were due to this type of virus. And this is just an influenza. Just as a note, it was named the Spanish flu because during World War I, the Spanish were neutral. And so everyone didn't want it to be named after them. So they named it after the Spanish. Just as a note, it didn't arise in Spain or anything like that. It's just named after the Spanish because they were neutral during World War I. But it was a really deadly pandemic. And you can see sort of through history, we've had multiple different strains of flu kill millions of people. And now we have sort of this new respiratory virus in COVID-19 that's on track to kill, depending on how fast we can get it under control, potentially millions of people. So viruses can be really, really deadly throughout history. In addition, HIV is sort of the new, relatively speaking, the new virus on the block, and it's been very, very deadly. So you can look at the time sort of in years, so starting in the 70s and 80s, and you can look at sort of how many people have died of HIV through time, and you notice that it's just going up in most countries. You're starting to see it starting to come down in the United States. That was in the 90s. It's coming back up again as new strains of HIV have evolved. But HIV has killed millions of people, and it's a particularly very deadly virus. It's something that, if you get it, you're pretty much stuck with it for the rest of your life, which is sort of an unfortunate thing. But what you will notice is that in areas like sub-Saharan Africa, where it is really, really prevalent, it has shown no sign of really slowing down over time. In addition, we're starting to see a rise in measles, and so here in Massachusetts, we saw a breakout of measles. In Washington State, they've seen breakout of measles. They've seen breakout of measles in Europe. They've seen breakouts of measles everywhere. And measles is a really infectious disease. When sort of measles first came to light, it was killing tens of millions of people each year. It was a really deadly disease. But we sort of got it under control with the vaccine for measles back in the 50s, which was, as a note, was developed at Boston Children's Hospital, in case you didn't know that. But sort of this surge in anti-vaccination has led to measles making a comeback. And we're starting to see countries lose their status of eradication. So, for instance, the United States, previous to the past couple of years, was eradicated in measles. We had no new cases of measles coming from within the United States. We would only get measles from people coming back from countries that still had measles. But because of this sort of anti-vaccination movement, we are no longer eradicated locally in the United States. And that sort of poses a problem because, as I mentioned, measles is a really deadly disease. Sort of just to wrap up this lecture and just to sort of tie it back to our lectures previously in this week, viruses evolved, right? They have genetic material, they can physically evolve. And so just to put this in perspective, I'll just talk briefly about HIV because it is one of the most heavily studied viruses on the planet. HIV has evolved multiple times. There are multiple different strains of HIV and they have varying level of deadliness. And so for instance, HIV-1 and HIV-2 are sort of the broad categories of HIV. And HIV-2 is pretty much endemic to Africa. It has a very low infection rate. It has a very low death rate as well. We believe this HIV-2 has been hanging around in Africa for about 100 years now. We think HIV didn't really emerge in the 80s. It emerged in the 20s and 30s, just as a note. But this HIV-2 evolved, it mutated, and it formed HIV-1, which is the globally pandemic strain of HIV. And within HIV-1, there are multiple subgroups, including a newly discovered subgroup called Group P, which is the deadliest known HIV to date. It can kill a human within 5 years or so, which is sort of in contrast to these other types of HIV, which take decades to kill you. Ultimately, there are, within sort of these groups of HIV, there are many different subgroups, and they're just named letters. And this is all through evolution. This is just random mutation happening inside the HIV genome. And ultimately, when we're thinking about the evolution of viruses, what's selecting for a virus to keep a mutation is really how good it is at infecting the host. Because remember, a virus is basically all it is a parasite, right? It infects you. And if it can better infect you or if it can better make copies of itself, that will be a trait that nature selects for and allows to proliferate in the population. And as it spreads from human to human, that trait will proliferate in the population as well. In addition, we can actually look at sort of an evolutionary tree of viruses, and we can look at HIV-2 and HIV-1. They're very distinct, but there are, in nature, there exists the chimpanzee immunodeficiency virus, which is CPZ, and we also have the simian influenza virus. And it's sort of interesting that the simian influenza virus is very similar to HIV-2, which implies that these share a common evolutionary ancestor, meaning that the simian influenza virus is likely the evolutionary ancestor of HIV-2. However, you'll notice is that the chimpanzee immunodeficiency virus is actually really closely related to HIV-1, which implies that we got HIV-1 from chimpanzees. And just as a sort of a last note about sort of HIV, how we got it, the way we got HIV is actually the same way we got COVID-19. People, particularly people that are in poor, less developed areas, eating bushmeat, being constantly exposed to these viruses, which allow these viruses to jump from one host to the next. Because one thing I unfortunately neglected to mention is that viruses have what's called a narrow host range, meaning that if I have HIV-1, it really only infects humans. It really can't infect any other species. And this is true for pretty much every virus on the planet. If you have one virus, it typically only infects one species of organism. And this is because the fact that every organism at the cellular level is very distinct. And remember, viruses are acting at the cellular level. Now, and so the idea being is the more and more you come in contact with viruses that can only infect another species, the more likely they are to jump from one species to the next. And so every time you hear about swine flu or bird flu, that's just because of extended contact with birds or pigs. So the more and more you're in contact with animals, the more likely you are for that virus to jump from that host to you. and it's more likely to evolve the capacity to infect you. So this sort of jumping from animal to human is really common. Most of the really deadly viruses that we know of, measles, HIV, COVID, all these different types of flu, jumped from continued exposure from animals to humans. So it's a really common route. It takes time, but these viruses, again, are evolving in real time to infect, say, pigs to now being able to infect us. So anyways, so that's going to sort of wrap up our talk about viruses. Oh, one last thing about HIV. There's this stupid, like, rumor that goes around that HIV arose through people having sex with monkeys. That's not true. That's never been true. It's sexually transmitted now, but it caught from eating bushmeat. It's not a sexual thing. Just to put that out there. I have someone ask me every semester. None of you have asked me, which I think is good, but just to put that out there, just to dispel stupid things. So anyways, with that, that's going to be sort of the end of our talk about microorganisms. I guess it's kind of good I didn't have any stuff on COVID because I ran out of time. As you can probably tell, I get really passionate about microorganisms because this is what my doctorate is in. This is what I'm most passionate about. So if you have any questions about anything about you saw today outside of the human microbiome, because we're going to have a class solely dedicated to the human microbiome later in the semester, please reach out. I'm more than happy to answer these questions. You want to talk on Zoom? I love talking about microbes. And I can see someone in the chat actually caught swine flu, which I feel kind of bad for you. That was a really nasty flu. I also see that I've increased the anxiety of microbes. I'm sorry. I was hoping to go the exact opposite route. I was hoping to sort of show you microbes, how important they are. And I think maybe later in the semester when we talk about how important they are to human health and your well-being, maybe that'll ease your anxiety a bit more. But, historically speaking, microbes are bad. And, you can look at all these people that have